So question 7. So it's talking about virus, okay? So let's look at question number part 1. Explain why viruses are not included in the 3-domain classification system, which is the archaea, eukarya, and bacteria, right? So why are they not classified? A lot of students write, oh, because it's non-living. The answer is because it is acellular or it doesn't have a cellular structure. Acellular. So you remember I teach you, we learned that life on Earth starts with a very simple unicellular organism. So we're talking about a cell. And then we also talk about as time passes by, the cell becomes more complex. So from a prokaryotic cell becomes a eukaryotic cell where there's membrane-bound organelles and nucleus. Then after that, it becomes even more complex because there's a compartmentalization where the cells will group together to form tissue organ, and system. So we're talking about cells, and based on the arrangement of the cells, then we start to group them into either of the 5 kingdoms. So here they say why viruses are not included in the 3-domain of classification is because it is not a cell, or it doesn't have a cellular component. That's the answer. So if you write it's a non-living, then there's no mark, because there is a statement here that says it is non-living. Discuss whether virus should be considered as living organism, and then student going to write, oh, because it's non-living. So we will not give mark for living or non-living. We are looking for acellular or no cellular structure. Okay, so this one you must remember. State the features that are used in the classification of virus. So answer must be based on the syllabus. So the syllabus says that a student must learn that a virus can be classified based on the type of genetic material, whether it's DNA or RNA, and also whether this DNA or RNA is single-stranded or double-stranded. So that's in your syllabus. Okay, of course, there's some way of classification where they classify based on the disease caused by the virus. For example, virus that can cause flu is called rhinovirus. So we classify based on the type of disease that they cause. Okay, so but this is not in syllabus, so therefore the one that we are looking for is this 2. But if you happen to write this one because it's also correct, you also get the marks. Okay, part 3, discuss whether viruses should be considered as living organism. So the key word of this question is discuss. Discuss means virus, you can say virus is non-living, virus is also living, so you have to discuss. So that means in the answer, I'm looking for both types of answer. It is non-living because of what? So you state because CAU. And then it is living because what? So you must have got these 2 to score full marks. So look at the mark scheme. The mark scheme says that you see, non-living maximum 3 marks, living at least 1 mark. So strictly speaking, to get the full marks, which is 4 marks, you must have got both non-living and living to score. But I didn't mark based on that mark scheme. If I mark strictly, there'll be more students that cannot score the marks already. Okay, so remember, discuss means write both ways. It is and it is not because of what. Okay, now first, it is not living. Non-living means to tell me because it doesn't have got the properties of living organism. So you tell me what are the properties of living organism. Living organism will carry out metabolism. For example, production of biochemical is part of metabolism. It will carry out respiration. It can reproduce. So they reproduce, the cell reproduce by replicating the DNA. Okay. And then no assimilation, no excretion. So all these are properties of living organism. Can assimilate means can take assimilation. Can take simple molecule and make it into complex molecules, so assimilation. Excretion, removal of waste, metabolic waste. Growth, locomotion means can move, and then can respond to stimulus. So all these are properties of living things. So the virus do not have this, that's why it's called non-living. So if you say virus can be considered as living, it's because it got some properties of living organism. For example, it is dependent on other organism. All living things on earth will depend on other organism, and they are linked together in the food chain or food web, right? For us, in order to survive, we need to eat, right? So we need to eat what? Vegetables, organism. Fish, so organism, right? Chicken, meat, etc. So we are relying on other organisms. So same as for viruses. They rely on other host cells to produce them. Then viruses contain genes, just like living organisms contain genes. Virus genes are used to code for new viruses. Okay. Virus have niches, so every organism got their own niches. Means all the factors that are required for the organism to survive. Okay, and all the roles that they do to their environment. So viruses have their own niches because they have got certain host cell that they will infect. For example, HIV will only infect helper T cell. So this is the niche of this HIV virus. Okay, and virus can evolve like living organism. So we know that no COVID virus from the year 2000 January until now, there are already a few different type of strains already, right? Because of mutation. So they can evolve because of mutation. Evolve means becomes different. Why become different? When phenotype is different because genotype is different. Why genotype is different? Because of mutation. Okay, genetic variation arises because of what? Many reasons, one of them is mutation. Okay. Can it be meiosis? No, because virus got no meiosis. Can it be migration? No. Only mutation. Okay? Yeah, migration can. Genetic variation, yes, you can, provided that already exists in other populations and then they just migrate. Okay, from China, fly to Malaysia or to Hong Kong, and then to US, and then fly to Malaysia. Can. Okay. Right, the next question asks about ecological sampling, right? Outline how an ecological survey can be used to measure the biodiversity of a terrestrial habitat. Measuring biodiversity. So we already learned that biodiversity can be studied in 3 categories, right? 1, 2, 3. So it's either ecosystem diversity or species diversity or genetic diversity. So if we are supposed to go to a terrestrial habitat like a jungle, in order to measure the biodiversity, usually among these 3 definitions, we will actually measure this one, species diversity, because it's easier for us to measure species richness and species evenness. That means we have to know how many number of different types of species that we have here, and for each species, how many number of individual for this species. So this can be done by ecological sampling. We can do a sampling. So outline how we conduct our survey in order to measure biodiversity. So remember, when you learn about ecological sampling, I need to retrieve the, so SMP is caused by a base pair substitution. A single nucleotide polymorphism is caused by a base pair substitution, mutation in a specific region of DNA. That means it's one particular base is being substituted. So for 2 strands, that means a pair, a base pair is being substituted. One method of identifying whether two individuals have the same SNP is to, so they want to see whether these two species have the same SNP or not. That means, are they very closely related? If they're very closely related, they will have the same SNP because they inherit from the same ancestor. But with time, they may not have it anymore because it get mutated into something else already, right? So use a specific primer and polymerase chain reaction. So these are the things that they do: add restriction enzyme, carry out electrophoresis, stain with a dye to compare the bending pattern. So these are all done in sequence. Make sure you know how to describe this in sequence, just in case you're asked to do a gel electrophoresis for maybe genetic fingerprinting, or maybe to identify the insulin gene from all the DNA that we have, okay, or to identify certain presence, certain allele that can cause a disease. So you need to state in sequence every step involved in this, okay? So this one is PCR, isn't it? So the question asks the student to explain. So why do we use primer? Why do we use restriction enzyme? And then also, why do we carry out gel electrophoresis? 3 marks, right? So it is when I see primer that a lot of students still do not understand the use of primer. So it's very important that you know the function of primer. Okay, so you learn primer when you learn chapter, I don't have it here, so I have to open it. Okay. Explain the difference between the probe and the primer. So you can see on the left side here, this is actually the probe. On the right side here, this is actually the primer. Their structures are similar because they're all single-stranded DNA, which can bind to its complementary strand, but their function is different. Okay, so probe. So the probe is always labeled one. So you can see here, this is called radioactively labeled probe. So the probe is a short length of single-stranded DNA that is used to attach to one end of a single-stranded DNA, and it is labeled either using radioisotope or fluorescent stain, so that the position of this piece of DNA after gel electrophoresis can be located. That means we want to know the presence of this particular allele, this DNA fragment, this gene, this VNTR region after electrophoresis. So which band of the bending pattern contain the DNA that we are looking for? Okay. Now, what about primer? Primer is a totally different thing. Even though structure is the same, it's also a short length of single-stranded DNA, but its function is to attach to one end of single-stranded DNA for the purpose of DNA replication. Because DNA polymerase can only lengthen a short length of single-stranded DNA. It cannot add nucleotide to a nucleotide. It must add a nucleotide to a polynucleotide. So the primer will act as a polynucleotide for DNA polymerase to add in the complementary nucleotides. So this is needed during PCR, polymerase chain reaction, to replicate or to amplify the DNA. So this one, please make sure you know. So that's why it comes out in the trial exam, right? Primer. So let's look at the mark scheme. So primer is used to bind to the target region, which is not sufficient, because, so what's the difference between primer and what? Primer and the probe. So you have to specify. And it is used for DNA replication. So this one is what you need to know. You need to mention. Or at least for DNA polymerase to bind to the specific complementary region to initiate DNA replication or to make the complementary strand. Okay. So what about the function of restriction enzyme is used to cut the DNA at a restriction site? Now, I do not accept recognition site because restriction enzyme bind to the recognition site. So it's supposed to bind to recognition site, but it's supposed to cut at the restriction site. So meaning to say that if this is a piece of DNA double-stranded, okay, this double-stranded DNA, okay, so this is a region where it binds is called the recognition site or binding site. But the part that it cuts exactly, let's say it cuts here. Okay, it cuts here to produce sticky ends, okay? So this part that it cuts is actually called the restriction site. So therefore, I'm very particular about using the word accurately in the right place at right time one. Cannot simply use one, okay? So therefore, this is not acceptable for me, even though I've marked some students correct, but strictly speaking, it is not correct. It is not correct. It's not accurate because you have learned a more accurate term than this, then you need to give a more accurate term. And this one is partially correct and partially wrong. Is it partially correct, partially wrong? Cut. Okay, not so accurate lah. This is accurate. Okay. Gel electrophoresis is used to separate the DNA fragments into its length order, right? So the smallest one will travel the furthest away from the negative electrode. Okay. Part 2. So the method of identifying whether two individuals have the same S&P method was carried out to compare species A and B. So this is when I think a lot of students read this figure 8.1 wrongly. Okay. So figure 8.1, you check carefully. Yeah. Now, can you see they label this thing as well? So the well is the location in the gel where I will load my DNA fragments. So I cannot consider these two as a bending pattern. This is not part of the bending pattern because this is the well where I load my DNA solution containing the DNA fragments. So therefore, some students' answer has actually considered these two as part of the bend, and they say that there's a similarity between the bending pattern of A and B, which is not correct already lah. So therefore, whenever you are given a figure, please read every single word that they label in the figure. Read this one, read this one, read this one, read everything first before you answer the question. Okay? To prevent writing the wrong answers and making the wrong assumption. Okay? So the question asks the student to describe and suggest an explanation for the results obtained in figure 8.1. So figure 8.1, you see, A only got one band, you know. B got two bands. So if A got one band, that means to say that we have added restriction enzyme. It seems that the DNA are not being cut. And because of that, the original DNA remain as a single, remain as one DNA, one piece of DNA is all moved together to the same location. There's only one band. So that means so first you describe, right? Then you explain. So describe is that in A, there's only one band. In B, there are two bands. Okay. And then you explain why is it one band? Because it doesn't have the restriction site for the restriction enzyme that's being used to cut the DNA. And then for B, there are two bands. That means to say that there's one restriction site, because in one DNA, when you got one restriction site that cuts, it gives you two fragments. So that's why I got two bands. Okay. Then because you've cut them, these two bands will be shorter than the original band. So the original band is longer, so it's located nearer to the negative electrode because the distance moved is less. Whereas these two that are being produced after cutting is smaller than this one. So they will move faster and further away from the well, which is negative charge. So they'll be further away. So that is another explanation. Okay, so 2 marks. Let's look at the mark scheme. Describe. So 1 versus 2 fragments. Explain. 1 fragment because no restriction site. Oh, there's one more mark. The 2 species do not share the same S&P. They don't have the same S&P. So what happened is that if you can see the band, that means to say that they do contain the S&P. Yeah? When you can see the band, you can see it because do we label them or not? Where do we cut them? So what is the S&P method? we have an SMP method? So this method is called SMP method, right? We use a specific primer. So we multiply the DNA. We add the restriction enzyme to cut at a specific site, and we carry out gel electrophoresis. And then we stain with the dye to compare the bending patterns. That means all the DNA fragments will be seen because we're not using a probe. We only use a stain. So the stain will stain all the DNA. So they say that both the DNA will not have the same SNP. This one can see me from here. How to see you don't have the same SNP? Because I didn't use any probe to identify SNP. So how do I know that whether they have the same? I can't. I disagree with the mark schema. I disagree with marking point number 3 because I can't see. There's no way for me to know whether there's SNP because I'm not using the probe for SNP to identify which one got SNP. So therefore, I disagree fully with the answer. That point, no, can't. Okay. So I still prefer to answer of why this one is located there because it's shorter, why this one is located here because longer, full stop. Okay. I think that one is a better answer. Right. Do you agree with me? You must know your stuff very well, then you can actually challenge the mark schema. Right. So yeah, sometimes I don't 100% agree with the mark schema because I know it's not accurate. Yeah. So of course, first thing is you must know your stuff first, then only you can no, actually challenge that answer. So next question, to identify the function of a gene, scientists can insert a copy of gene to plasmid to create recombinant DNA. Plasmid transfer the host bacteria to express the gene. Okay, very good. Refine the recombinant DNA. Even this answer also a lot of students cannot score. So that means the student have not studied this part, or that's the reason. Okay, so let's look at the mark schema. So what is a recombinant DNA? Now see the keyword is organism or sources. That means this piece of DNA is made up of DNA that comes from 2 different organisms or 2 different sources. So this is the answer. Okay, 2 different organisms. Like for example, plasmid is from bacteria. Insulin gene is from human. So human insulin gene plus bacterial plasmid becomes a recombinant DNA. Why? Because these 2 DNA comes from 2 different organisms or 2 different sources. Okay. Right. So next question. So there's a plasmid that's given here in a diagram, figure 8.2. So this plasmid includes a gene that codes for fluorescent protein GFP. So you can see that GFP is located here, right? Okay. So other than that, we have got what else? We have got this gene for antibiotic resistance. We have got promoter region. For the promoter region is for the DNA polymerase or the RNA polymerase to bind, right? POS. And then multiple cloning site, which we have not learned before, right? So what do the question ask? Explain the purpose of including a gene for fluorescent protein in the plasmid. Are very interesting. So we already learned that GFP is a marker, right? Remember? The word marker denotes the gene that we have added in to actually identify whether a bacteria has taken up the recombinant DNA, because that recombinant DNA, the plasmid also contains the marker. So we want to identify the transformed bacteria. That means the bacteria have taken up the plasmid or not. And how do we identify? Because the marker gene will express itself and produce the GFP, right? GFP. And then GFP will fluoresce under the UV light. So the bacteria will also fluoresce. So this is how we identify. Okay. Next question is, name the enzyme that's used to join the cut ends of a gene. So this one is very easy, DNA ligase lah. Okay, DNA ligase. So that's all.